Hello guys, in this part I want to talk about the immune system. And first I want to introduce the infectious agent to you. The infectious agent are organisms that can cause damage or possibly death to the host organism that they invade. Infectious agents that cause harm to the host are said to be pathogenic. The five major categories of infectious agents that cause disease in humans are bacteria, viruses, protozoans, and multicellular parasites. And when we talked about the 11 organ system in 2050, we don't talk about the immune system to you. This is because unlike those 11 body systems, the immune system doesn't have organs. Instead, numerous types of cells, secreted molecules, plasma proteins can work for our immune system. The function of the system is to protect our body from infectious agent, and potentially they can invade and remove harmful substances by providing immunity in our body. And in this chapter I want to talk about this immunity. The function dependent on a specific type of infectious agent. For each infectious agent we have the specific type of immunity in our immune system and in our body. The first cells which are very important for our immune system are leukocytes or white blood cells. And you know that they are a specialized cell in our immune system. They formed in the red bone marrow. Some of them come from myeloid stem cells, and some of them are from a lymphoid stem cell. We have granulocytes in these categories, and the granulocytes are divided in three parts: neutrophil, eosinophil, and basophil. We have monocyte, which is the agranulocyte cell, and most of the times the monocyte cannot live in our blood for the long time, so they come out of the bloodstream, they go to connective tissue, and when they go to connective tissue we call them macrophages. And the last cells, which are the important cells in our immune system, are lymphocytes. And we have three types of lymphocyte: B lymphocyte, which mature in our bone marrow; T lymphocyte, which mature in the thymus; and natural killer cell or NK cell. They are not B cells, they are not T cells, they are about 5% of lymphocyte, and all the times they monitor our body. They are wandering cells in our body and try to find tumoric cells, pathogens, and unwanted material in our cell and destroy them. Also, we have some structures which can host immune system cells. Most leukocytes are found in our body tissues. The primary location of them are the lymphatic tissue, and I talked about lymphatic tissue before for you. I told you that we have lymph node, spleen, tonsils, mold, and lymphatic nodules. And in these organs or in these tissues we have T lymphocytes, B lymphocytes, macrophages, dendritic cell, and natural killer cell. And some of them are located in our blood. And we have some selective organs which can house macrophages for us. I told you that the monocyte cannot live in our bloodstream for a long time. When they come out of the blood, we call them macrophages. And in each part and in each organ of our body, the macrophages have different names. For example, in our bone, we call them osteoclast. In our lung, we call them alveolar macrophage or dust cell. In the skin, we call them dendritic cell. In our brain and the spinal cord, we call them microglia. In our liver, we call them Kupffer cell. They have different names, but all of them have the same function, and the function of them is doing phagocytosis. The other part of our body which can house immune cells are epithelial layer of the skin and the mucous membrane. They can house dendritic cell, and dendritic cell is the cell which derive from monocyte. We call them dendritic cell because they are branched and they are very similar to the dendrites of the neuron, and they can do engulfment of pathogens. And in the connective tissue, which we have beneath the epithelium, we have the other type of immune cell, which we call it mast cell. Some scientists think that the mast cell come from basophil, and some of them think they are the separate cells and they don't have any connection to the basophil. And they are located in the connective tissue. They are located very close to the small blood vessels. They can secrete histamine and heparin, and this histamine and heparin can make vasodilation and initiate the inflammation for us. We can see them in the mucosa or the epithelium of the respiratory system, gastrointestinal system, and urogenital tract, and we can see them in the connective tissue which wrap the organs, like endomysium or endoneurium. And we have a group of proteins, plasma proteins, which we call them cytokines. Cytokines are small soluble proteins which are produced by cell of both the innate and adaptive immune system. I will talk about the innate and adaptive immune system later for you. The innate immune system is the immune system which we have when we were born, and the adaptive is the acquired system which we can acquire it throughout our life. And these cytokines are produced by the cells of both immune and adaptive immune system to regulate and facilitate immune system activity. These soluble proteins serve as a means of communication between cells. They can control the development and behavior of the immune cells, and they can regulate the inflammatory response to the innate immune system, and they can function as a weapon to destroy cells. Cytokines have also recently been shown to influence other non-immune cells, such as those which we have in our nervous system. A cytokine is released from one cell and bind to the specific receptor on the target cell, where its action is similar to that of the hormone. Cytokines can act on the cell that releases it. It can act on the local neighboring cell, and also it can circulate in the blood to cause systemic effect. So it can do autocrine function, it can do paracrine function, and it can do endocrine function. They have a very short life. Although the terminology of cytokine continues to evolve within the discipline of immunology, a current method of organizing cytokines identified in different categories. We have interleukin, tumor necrotic factor, colony stimulating factor, and interferons. And they are the different classes which we have for cytokines, and I will talk about them later here for you. Their effect is they are signaling cells. They can control development and the behavior of the immune cells, and they can do regulation of inflammatory response, and finally they can destroy unwanted cells in the body. In this picture, you can see the location of the immune cell. If we focus on each of them, you can see them. The immune system are located in lymphatic tissue like lymph node, spleen, tonsils, and mold. In these tissues, we have T lymphocyte, B lymphocyte, macrophages, dendritic cell, and natural killer cell. We can see immune system in select organs like lungs, like skin, like liver, bone, and nervous system too. You can see The immune system, like dendritic cell in the skin and the mucous membrane. The mucous membrane is the epithelial tissue. And also we can see the immune system, the immune cells are like mast cell in the connective tissue in all parts of our body. And the most important part and location of the immune cell is in the blood. In the blood, we have granulocytes like neutrophil, eosinophil, and basophil. Also, we have agranulocytes like lymphocyte and monocyte. So they are the primary location of immune cell in all parts of our body. We have two types of immunity in our body. One type of immunity is called innate immunity, and one type of immunity is called adaptive immunity. The means of protecting the body through the cells, cytokines, and physiologic process of the immune system are organized into two categories of immunity. One of them innate immunity, and the other one is adaptive immunity. Although both work to protect us from harmful agents, they are different from each other in some factors. First, the cell which evolve, specificity of the cell response, mechanism of eliminating harmful substances, and amount of time for their response. So in these four different parts, they are different from each other, but the final result of both of them are the same. They want to protect us against different pathogens. Although innate and adaptive immunities are distinct, they work together in body defense for us. First, the innate immunity. Some defense mechanism of the immune system protect us against numerous different substances, and because we are born with these defenses, we call them innate immunity or non-specific immunity in our body. The components that provide innate immunity collectively form the innate immune system in our body. This includes the barrier of the skin and the mucous membrane that prevent entry of different harmful substances to our body. We have some non-specific cellular and molecular, which can fight with different pathogens like macrophages, and some chemicals like interferons, and some physiologic process like inflammation. All of them can protect us against different diseases. The response of our innate immunity is very, very quick, and it can respond immediately to potentially harmful agents. The other type of immunity is called adaptive immunity. This is the other defense mechanism of our immune system. And in these parts, we have T cells and B cells which can work against pathogens specifically. Each lymphocyte, whether a T lymphocyte or B lymphocyte, respond to a specific foreign substances or antigens to which we are exposed during our lifetime. The lymphocyte which can respond to chickenpox is different from the lymphocyte which can protect us against rubella or against tetanus or against rabies. This type of immunity provided by lymphocyte is referred to adaptive immunity or acquired immunity or a specific immunity. And the lymphocytes that are responsible for doing this immunity is called adaptive immune system. Lymphocytes can provide a powerful means of eliminating foreign substances. They cannot answer very quickly, so it takes several days to be effective. And in this chart, you can see the immune system, and we have two types of immune system. One of them is innate immune system, and the other one is the adaptive. The innate immune system can provide innate immunity. They have are multiple components that protect us against a wide variety of substances. We have a skin and mucous membrane, which can prevent entry of the pathogens. And also we have some non-specific internal defenses. We have some cells like macrophages and natural killer cell. We have some chemicals like interferons and complement, and we have some physiologic response like inflammation and fever. Pay attention, inflammation and fever are not disease. They are signs of presence of a bacteria, a virus, and pathogen in our body. So they are a process of immune system which can protect us. And the adaptive immunity is provided by adaptive immune system, the lymphocytes that are activated to do replication and respond when we have a specific pathogen. And their response has delay, so they cannot work immediately. We have T cells, which can do cell-mediated immunity, and we have B cells, which can do humoral immunity. The B cells should change to a cell, which we call it plasma cell, and plasma cell can make antibody and protect our body against antigens. In this part, you can answer these questions. Where are the mast cells found? In what ways do cytokines resemble hormones? Pay attention to their function: autocrine, endocrine, and paracrine function, and which type of immunity takes longer to develop. First, I want to talk about the innate immunity. I told you that the innate immunity is provided by the innate immune system, which include the skin and mucous membrane, which help to prevent entry of harmful substances, various types of cells, chemicals, and physiologic process. Within the body, they can provide non-specific internal defenses. If you imagine your body to be like a fortress, the skin and mucous membrane can provide it with its first line of defense, and the non-specific internal defense can provide the second line of defense. The second line of defense are these defenses: activation of neutrophil, macrophages, dendritic cell, eosinophil, basophil, and natural killer cell. We have some chemicals. I told you that we have interferons and complement, and we have some physiologic process like inflammation and fever. The few microbes can penetrate through our skin, but you know that the epithelial tissue have intercellular junction, and the first junction which we have between the epithelial cells are the tight junction or zonula adherens. And this it can make a physical barrier of the epidermis and the skin, and it doesn't let any pathogens to come inside. Besides this physical barrier of epidermis and dermis, we have some antimicrobial substances which are released from our skin. Dermacidin, lysozyme, and defensin are secreted from our sweat glands, and they can prevent overgrowth and multiplication of the bacteria on the surface of the skin. And also we have sebum, which is secreted by sebaceous gland, and it has also antimicrobial and antifungal substances. You need to know that on the surface of our skin we have a normal flora. Some bacteria live on the surface of our skin always with us, but their population should be in the constant level. If for any reason their population increase or decrease, then the invaders and harmful bacteria or viruses can penetrate through our skin and make problem for us. And this normal flora is very important for our skin. We have mucous membrane line the body opening, or the epithelium which line the body opening. You know that your mouth cavity, the vagina, and also the urogenital system, and also your respiratory system is lined by epithelium. And this epithelium doesn't let anything to go inside. This epithelial tissue can produce mucin, and they can release some antimicrobial substances like defensin, lysozyme, and IgA, which I will talk about it later for you. And then we have the line of harmless bacteria, which can suppress the growth of more virulent types of bacteria. The cellular defenses which we have in our innate immunity are neutrophil, macrophages, and dendritic cells. They can do engulfment of unwanted substances by doing phagocytosis. The neutrophils and macrophages can destroy engulfed particles for us. They intake vesicle fused with lysosome and form phagolysosome, and I will show you this in the next picture. Look at this picture. Here we have one unwanted particle or infectious agent. The macrophages or the neutrophil can make pseudopodia around it and do endocytosis or engulfment. When it comes inside the cell in the vesicle, we call it phagosome. The phagosome and the lysosome of the cell, which contain digestive enzyme, combine together and they can make phagolysosome. The digestive enzyme can destroy and it can break the pathogen to small pieces, and after that this residue and the small particles can do exocytosis and come out of our cell. This is the function of the macrophages, and the dendritic cell is the name of the macrophages which we have in the epithelial tissue or in the mucous membrane. So they can work like this to destroy the pathogens for us. And we have a process which we call it a respiratory burst or oxidative burst. The release of some molecules like nitric oxide, hydrogen peroxide, and superoxide. All of them are reactive oxygen containing molecules, and they can do respiratory burst, or they can destruct the bacteria and viruses. The other cells which we have here is a dendritic cell, and the dendritic cell can destroy the particles, and they can present the fragments of the particles on their surface. We call this dendritic cell antigen presenting cell. Look at here. Imagine that this is the macrophage or dendritic cell. It destroys the pathogen, and the particles of the pathogen come out of the cell by exocytosis. Some of these particles can attach to the surface of the cell. When they attach to the surface of the cell, we call these macrophages APC, or antigen presenting cell. This antigen on the surface of the macrophages is a sign, and it can help our immune system to increase the specific number of our leukocytes. For example, if the pathogen which we have here is the parasite, the particles of the parasite come to the surface of the macrophages, and when these pathogens represent to our immune system knows that it should increase the number of eosinophil. If we have bacteria here, the particles which are here can stimulate our immune system to increase the number of neutrophil. If we have virus here, Then the particles of the virus on the surface of the cell stimulate our immune system to increase the number of lymphocytes. So we call these macrophages or dendritic cell APC or antigen presenting cell. They present the specific type of antigen to our immune system, and then our immune system increase a specific number of leukocytes in our blood. And the other cells are basophil and mast cells. These two cells can promote inflammation for us. The basophil circulate in our bloodstream, but the mast cells are located in the connective tissue, in the mucous membrane and internal organs, and most of the times they are very, very near to the blood vessels. They release some granules, and these granules contain some chemicals. The chemicals which are secreted here are different. We have histamine, we have heparin. The histamine can do vasodilation and they can increase the permeability of the capillaries. The heparin works as anticoagulant and it doesn't let coagulation and blood clotting happen. And both of these cells can attract the other immune cells. This attraction has a specific name in immunity, and we call it chemotactic. And also the basophil and the mast cell can release a chemical which we call it icosanoid. Icosanoid released from their plasma membrane, and this icosanoid can increase the process of inflammation. In this picture, you can see the function of basophil. The basophil can release histamine, and the histamine is a vasodilation agent, and also it can increase the capillary permeability. So when the capillary permeability increase, more particles, more leukocytes can come out of the bloodstream, and also more plasma can come out of the blood vessels. The vasodilation can make redness and also the increase the temperature of the affected part, and all of them are the signs of inflammation. When one part is inflamed, you can see redness, you can see that part is warmer than the other area, and you can see the swollen part, and all of them are the function of histamine here. The heparin can do anticoagulant activity, and also we have icosanoid which can increase the process of inflammation. In our immune system, the other cellular defense which we have in innate immunity is the function of natural killer cells. They can destroy a variety of unwanted cells. The natural killer cell form in our bone marrow, it can circulate in the blood, and then it comes out of the blood and accumulate in the secondary lymphatic structure like lymph node, like spleen, tonsils, mold, and lymphatic nodule. And they can do immune surveillance. They can patrol the body detecting unhealthy cells. They destroy virus-infected cell, bacteria-infected cell, tumor cell, and the cells of transplanted tissue. So when we have graft, these natural killer cells go there and they start to destroy the graft cells. They kill the cells, tumoric cells, or virus or bacteria affected cells by releasing cytotoxic chemicals. We have two chemicals which are secreted from the natural killer cell. One of them is called perforin, which can make pore in the cell membrane, and the other one is granzyme. And the granzyme can enter the pore and it can cause apoptosis in the cells. Apoptosis is the cell death. Look at here. In this picture, you can see the function of natural killer cell. This is natural killer cell. It can secrete perforin. Perforin is a protein which comes and it's inserted to the cell membrane, and it can make pores in the cell membrane. And through these pores, the other particle, which is granzyme, come inside, go to the nucleus, and make apoptosis of the cell. And you see that if this is a tumoric cell, if this is a cell which is affected by bacteria or viruses, they start to die. And the other cell which we have here is eosinophil. The eosinophil can attack multicellular parasites. They can do degranulation, they can release enzyme and other toxic substances. They can release some proteins which can make transmembrane pore in the parasite cell. They participate in immune response of allergy and asthma, and they can engage the phagocytosis of antigen antibody complex. Look at here. In this picture, you can see the function of eosinophil. The eosinophil can make some cytotoxic chemicals. These cytotoxic chemicals can make some pores on the surface of the parasite. And then through these pores, imagine that we have some pores here. And from these pores, the fluid come inside the cell or inside the parasite. The parasite become bigger and bigger, and then it bursts. And this is how the eosinophil can destroy the pathogens and the parasites for us. The fluid which we have in our body can protect us. We have lacrimal apparatus or tear gland in our eye. It can produce tear, and tear contains many lysozymes, and also it can respond to irritation. If anything comes to your eye, the tear is secreted and remove the item, that particle from your eye. The tear contains lysozyme, and the lysozyme can do digestion of the pathogen. We have saliva which can wash microbes from our teeth and mouth. You see that the animals lick their baby because in their saliva they have lysozyme and they have different enzymes which can break down the pathogens. We have a urine secretion with the acidic pH, and it can clean the urethra, and it can remove all pathogens based on its specific acidic pH. We have vaginal secretion which can protect the female genital system, and we have defecation and vomiting. Both of them are the process which can remove microbes from our body. In our body, we have some chemicals which can protect us, like sebum, which can make a protective film on the surface of the skin. The sebum contains antimicrobial material. If your sebaceous gland stop working, your skin become dry, then you have some cracks on the surface of the skin, and the microbes which you have on the surface can easily come to your skin and make infection. We have perspiration or sweating which can flush away microbes. We have gastric juice with the acidic pH, and it can destroy microbes by the acids. In our body, we have different types of cytokines. I told you that. I told you that the cytokines are small soluble proteins which are made by innate and adaptive immune system, and it can regulate and facilitate immune system activity. I told you that they serve as a means of communication between cells. They can control the development and behavior of our immune system. They can regulate the inflammatory response, and they can function as a weapon to destroy cells. And we have different categories for cytokines. We have interleukins, we have tumor necrotic factors, we have colony stimulating factors, and we have interferons. They are different, but all of them have the same function. They are released from one cell, bind to the specific receptor on the target cell, and then they start to destroy the cell by different mechanisms. Sometimes they can do autocrine stimulation, sometimes they can do paracrine stimulation, and sometimes they can do endocrine stimulation. The interferons is one of these cytokines, and they can work not specifically against the different viral infection. This interferon is secreted by macrophages, lymphocytes, and fibroblasts. It's released after viral infection. They include antiviral protein and they stop replication of viruses in our body. The other antimicrobial substance which we have in our body is complement. The complements are inactive protein in our blood plasma, and they are located also in the plasma membrane of the cells, and they can make cytolysis. Look at this picture. When we have bacteria or parasite in our body, the complement, which are inactive protein in our blood plasma, come and insert themselves in the cell membrane of the bacteria, as you see here, and they can make some holes on the surface of the bacteria. Through these holes, the fluid and salt can come inside. The bacteria has a capacity to hold this fluid and salt, so it becomes expanded and then it bursts. So this is the action of our complement system against bacterial infection. The complement system have two different processes. One of these processes is called opsonization. Opsonization means the complement protein, which we call it opsonin, binds to the pathogen. Look at this picture. In this picture, you see that this complement, which is secreted by macrophages or leukocytes, come and attach to the pathogen, and it enhances the phagocytosis of this pathogen. And the other process is inflammation. Inflammation is enhanced by complement. This complement can activate the mast cell and basophil, and then it can attract the neutrophil and macrophages, and the inflammation starts in our body. The other antimicrobial substances are iron-binding protein. The iron-binding protein are the proteins which bind to iron, and the iron is very necessary for growth of bacteria. So one way which we can stop replication of bacteria is this. We need to remove iron from the axis of bacteria. Transferrin, lactoferrin, ferritin, and hemoglobin are the proteins which bind to iron. Remove iron from the axis of bacteria, and then the bacteria cannot do growth and multiplication. And we have some small antimicrobial proteins. They are very short peptides. They can kill bacteria, like dermacillin, which is secreted by our sweat gland, catalycidin, which is secreted by our neutrophil and macrophages, and thrombocidin, which is secreted by our platelet. And all of them can stop function of the bacteria. In this picture, you can see the process of phagocytosis. In this picture, you see that when we have a microbe, we have a process which we call it chemotaxis. From the microbes, we have release of some chemicals which can attract our immune system cell like magnets. So by this chemotaxis, the macrophages can come near the microbes. Then they attach together. We have some pseudopodia which is created here, and we have endocytosis or phagocytosis. The bacteria, the virus, or the pathogen come inside in the phagosome. Phagosome and lysosome combine together and make phagolysosome. The digestive enzyme release on our pathogen, and the pathogen is digested, is broken down to small pieces, and then these small pieces come to the surface of these macrophages and start fighting with these pathogens.